just go to the poster list and use this.
Ciao, Yellow. Hi, Yellow.
Ciao. Hi. Yes, yes,
Hi.
I do. Alice.
Well, in that case, I will be the first one to break the ice.
Thank you.
Good morning to all from Serbia, from Belgrade. It's although yesterday, hi, Professor Tsenkova, nice to see you again. I really deeply congratulate to you for the organizing of this wonderful conference, wonderful event. Although the situation is challenging, but somehow we are succeeding to increase the collaboration, well, to emphasize what is important, not only in the field of COVID-19, but also some other important topics which will remain after this pandemic be behind us. So although the spring started yesterday on our planet, this morning is snowing in Belgrade. So Yellow, just to tell you that we have it's not a big snow, but it's a snow. So I would like to congratulate to you all for the organizing this event and to say that I'm really happy to be the part of this conference. So my poster is available. I'm not from the field of aquaphotomics, slightly moving to this field, but from the field of environmental biotechnology, and I will gladly answer to all the questions regarding perfluorinated compounds today and aquaphotomics, with a little help of my friend Jelena.
Yes, of course. So I'm going to project the screen if needed. But we had I mentioned the poster yesterday when I was talking during the workshop and during my talk. Any question regarding Vlada's poster? And for me also, I guess. Share the screen.
Can you, yeah, okay. You're going to share the poster,
yes,
right?
yes.
Well, in that case, before you share, I mean, during sharing the screen, I would like to emphasize that these chemicals are very interesting. These perfluorinated chemicals become interesting in Japan some 12, 15 years ago. But and unfortunately, not so much worldwide. But last year we had a huge project call titled Green Deal, and it was the call from the European Commission, and that the value of the whole Green Deal is 1 billion euros. Some 1/5, around 200 million euros, is devoted to solve the issue of the perfluorinated compounds in the environment. So now that topic is starting to be really important, well, in Europe and worldwide. United States EPA also announced a call regarding, in particular, these compounds. So what we try to do, we try to see if it is feasible, if it is possible to analyze the perfluorinated compounds using aquaphotomics, and to see if there are any change in a pattern of the water molecules when these chemicals are dissolved in the water. And if I remember good, Yellow, we saw some changes.
Yes, we absolutely saw the changes. Not only I can't manage to share my screen. I'm sorry.
Is it okay if I share my screen?
Yes, please, if you can. Yes.
Okay. Just to so, sorry.
Yeah.
Well, I hope that now you can see the screen.
Yes, just a little bit big. Okay.
Oh. Yeah. It's so maybe I can reduce the size somehow.
Yeah.
So the question is actually, is aquaphotomics suitable for the determination of an analysis of the perfluoroalkyl substances? And there are around 4,700 different compounds, but PFOS and PFOA are now on the list of the Stockholm persistent organic pollutants compounds. And these compounds are known as forever chemicals. What means forever chemicals? Well, they will remain in the environment forever. However, you know, I'm from the field of environmental biotechnology, and there are some evidence that microorganisms can conduct some type of defluorination of these compounds with not much success, like when we are talking about petroleum hydrocarbons, but also some plants can conduct biosorption of these chemicals. The basic idea here was to see if there is a change in a water pattern in the near infrared when these chemicals are dissolved in the water. Or the question is, can we use aquaphotomics to analyze water, to screen, not to determine the exact concentration of these chemicals, but to see if we can use aquaphotomics to determine if these compounds are present in the water. And Jelena, if you can tell me, so can we use aquaphotomics?
Yes, thank you. Thank you, Vlada.
Maybe it's difficult.
No, it's not. So it's not that we can detect concentrations in parts per billion and so on, exactly the number, how much there is. But the point is that in both cases of these compounds, we detected a very similar pattern of water which corresponds to surfactants, to these kind of compounds. So I think that is very good, because the range which we worked with is parts per million and parts per billion. And we also managed to connect those results with other substances, not only pollutants, but something that can be emerging pollutants.
Yeah, thank you so much. So actually, these results are very, very promising. What we can do in the future, we can also try to compare with some other surfactants. But this is excellent because now we have another method to analyze the presence of these compounds in the at least to screen the presence of these compounds in the environment. What is also important, since microorganisms can interact with these chemicals, I would so much like to see, the product of the microbial interaction with these chemicals analyzed by aquaphotomics.
Yes,
So
yes,
maybe
yes.
something like that we can do in the future.
Absolutely. So today's day, when we talked about the material water interaction, these guys were always on my mind because surfactants basically do not accept, material cannot attach to them, like in the Teflon dishes and so on. And I was really wondering then what can be used, because there are ways to adsorb these species and remove them from the contaminated, for example, waters and so on. And I know that there are some bacteria there that are being engineered genetically to actually pull this out. So I was wondering what kind of mechanism, what kind of biomolecules are used, and maybe something can be done. I would like that.
Perfect, perfect. So I don't want to take the stage anymore. Just to tell you, all the time you're next to me.
And you're next to Professor Tsenkova, by the way. Professor Tsenkova has your cup always. Thank you. Yes, so
kisses and hugs from Belgrade. This is like a Eurovision contest. I like this.
Exactly. Thank you. So Vlada, just make sure, Pierre, poster is uploaded. You can download it from the poster page, and you can always get it from me if you don't have it now. Now, I would like actually to ask if from Hungary, Flora Vitalis is here, and if she can project her poster, because I have a few questions. Flora?
Good morning, everyone. I am here.
Good morning.